So I'm not sure if you heard about Philosec, but this has been a prevailing format for microbiome data in R for the last decade, and this is a very good format, but there is now complementary format that extends that a little bit and provides capabilities for time series and multi-omics assays. And this we call tree summarized experiment. Here you see a workflow that comes from importing the raw data. So instead of importing raw data, we can also generate microbiome data, like Daniel does with all these different dynamical models. But essentially, we get some microbiome data, whether it comes from real experiments or simulations. We have that, and then we put it in a certain type of data object in R. I explain this in a little bit more detail soon, but then all the tools that you have in R can assume that the microbiome data is always in a particular format. The matrix is always like features times samples. The background information of the samples is given in a particular way. The same applies for the taxa and so forth. So then all the different methods can assume this format, and you can start to build interoperable methods that are very nicely forming an ecosystem of interoperable tools. So there we have the MIA pipeline, so you can feed in this data object into various tools for the analysis and visualization, and then you can have reproducible pipelines from the original microbiome data to the final analysis and visualizations. We formally introduced this in European Bioconductor meeting in 2 years ago, and there is a tutorial on this, and I will demonstrate a little bit how to use this with time series in particular, because this is the topic for the dynamical models. So let me go here. So the main idea is that the data class defines the data structure, and then there are the ecosystem of methods around that and the community of developers and users. So there is quite many packages in R available for not only for 16S analysis, but also for taxonomic profiles from metagenomes. And there is like dozens of R packages that are able to utilize different microbiome specific techniques. And the Bioconductor project itself, I think, deserves a little bit of explanation. So this is a project that has been running for 20 years. It's a global developer project for bioinformatics software, open source software. You can see a timeline of contributed packages here on the bottom right. So there is currently more than 2,000 high quality packages available. I say high quality because this includes software review. It is not that easy to get a package accepted in Bioconductor repositories. There is regular release cycles, so they all the time make sure that all the packages are passing various checks and unit tests. So there is continuous maintenance with all the new R versions and new versions of other packages that the tools depend on. So there is high quality control and good software review, and this makes it sure that you can trust and rely on these resources in the longer run. Of course, it always depends on the developers, but this is really like a community-driven project, and it is not limited to microbiome research. You also have many other areas of bioinformatics covered, and of course, often there are interesting connections. For instance, we often need to use functional analysis tools as well. And there is also material for time series analysis. So this tree summarized experiment container, I have to explain a little bit before the demo so that you understand the basic idea. So the data container starts with typical taxonomic abundance matrix, where you have the different taxonomic groups on the rows, and then you have samples on the columns. So in the case of time series, these columns can be samples from the same individual, but measured in different time points, or not necessarily individual. It can also be a synthetic community or simulated community, but the point is that it can also form a continuity, that all the samples came from the same community and they are just a development over time. And each time point will then have a profile over all the taxonomic features. So this is very typical taxonomic abundance matrix in microbiome studies. And when we do multi-omics, we then tie this with other omics. So we put that in the center of the 3 summarized experiment container as a matrix where the features or rows are the taxa and the columns are the samples, as I mentioned. But then in addition to this, we can have background information for the samples. So for instance, for each time point, we can have information on not only on time, but maybe also on some parameters of the host or environment, which can be quite important in multi-omics studies. We can have information on the pH, for instance, temperature, and different factors like this. So this is all background data for the columns for each sample in each column. And of course, this is highly relevant when we analyze the data. Then on the rows, we have the row data, which is background information for the taxa. And here we can have information, for instance, for the phylogenetic classifications for the taxonomic groups, if relevant. And we can also have a phylogenetic tree on the rows and all kinds of hierarchies, hierarchical information. This hierarchical information, we could also potentially have samples. So this is the basic structure. Then you also see some layers here. These layers are meant to be used for different statistical transformations. Maybe, well, I'm sure you are all familiar with logarithmic transformation. In microbiome research, we also have a transformation like centered log ratio transformation and other Aitchison transformations and other types of transformations that we often use. So we can include 3 different versions of the data in the same object as different layers. And this is useful when you do subsetting. If we want to subset to particular time points, for instance, then we will do the subsetting in single object for all the layers at the same time. So there are benefits like this in dealing with these data objects. And there are many demonstration datasets available, but here we have been using the MIA-Sim simulated communities. And let me see. If we look at some of the examples before I go to the demonstration, more clearly here on the screen. So this is just an example about the type of transformation we could have. So transform samples means that we are transforming samples with a particular transformation here. Centered log ratio transformation, we apply this to the 3 summarized experiment data object. We add pseudocount. We can also calculate unifrac distances, for instance, if we want to analyze how the time series diverges from its baseline. And now if we do these microbiome data simulations with the package like MIA-Sim, which Daniel already explained in a lot of detail, we can simulate data from these different communities. And then the output from the simulations will be readily in this useful format. So we get a data container out of the simulations, and then we can use all these different downstream tools to analyze and visualize this data object. And here you can see some time series that have been visualized by using these tools, and you see the code on the left. So this code first just uses the consumer resource model for the simulation with various parameters. But Daniel basically covered this already. And then in the last line, we basically plot this time series. So basically, you can use just one line command. Once you have a data in this particular format, you can just use one line to plot figure like this. So you do the simulations, then you can use one-liners to visualize the time series. And of course, there are many other handy tools. You can look at the MIA time package. The MIA time package is accompanying this MIA-Sim package. So whereas MIA-SIM provides tools for simulating the communities, the MIA-Time provides tools for the time series analysis of the communities and for the visualization. So we can look at the individual time series, and if we have community, we can also do more complex tasks. For instance, we can calculate divergence from baseline. So this is not just the community time series as abundances, but actually what you see here in this figure is the divergence from the baseline. That's the y-axis. So how different the time series samples are from the baseline when we do beta diversity estimation. And this happens to be a bioreactor example, and we can see that there is quite some divergence initially, and then the community will stabilize over time following some perturbations. This is an example of a recent study by our colleague Sudarshan Shetty. Here is a code. It's a bit small here on the left, but if you look at the website of the MIA-Time package, you will have this example and various other examples there that you can basically adapt. They are all reproducible, so if you have these packages installed in your R, then you can just try out the examples from this package. And then finally, there is the visualization package MIA-Vis. Here you just see some examples on running various visualizations for the community. And of course, you can do this for the simulated communities with MIA-SIM, but you can equally well do these things for real communities. So there are many different visualizations, of course, and we can have a look at some of these in more detail if you're interested, but this slide is just showing some variation, what is possible. Again, you can go to the website of the MIA-Vis package to have a closer look at examples of these. So this was a small introduction to this idea of these containers and the context, why we want to do these things in this way. The interoperability of the tools is the key. And before I go to the demonstration, I maybe wanted to ask if you have any kind of questions at this point about the material.
Leo, I think I have again a very silly question. That okay, I have seen in the previous slide that you subtitled self-organize instability.
Here or here?
Yeah, here. But what does it mean here?
I think this is, well, it's one type of dynamical model and that has particular properties. I think actually, Daniel, did you include this in your recent publication? Because I think this is more like a domain of yours. I can explain on a rough level what it is, but Daniel is, I think.
Yeah, it's like a Lotka. It's an individual-based Lotka-Volterra model. It's from it's a published model. So.
Some properties of it is like if you have a time series that follows this model, it can be kind of stable for a long period of time, but it gradually approaches a point where you kind of reach instability. Like if you imagine a pile of sand, for instance, where you put sand, you know.
I know this system. This is the reason I asked how is it connected with these physical systems. You know that maybe the reason is that microbial system, you know, modified the environment somehow.
Right. Actually, to be honest, I have not seen this used very much in the microbiome context. It is an interesting complementary model for community time series, but I have not really encountered many works that utilize this really in the microbiome works. Daniel, are you aware of such works? It is included in this package.
I think it's only one study. It's only one. I mean, there's this study from Caroline that uses this model, but the yeah, it's just the. I'll put it in the chat. It's just this one paper that made the model. People confuse it very.
Thanks a lot.
Yeah, so I think in practice, this Hubble's neutral model and generalized Lotka-Volterra models are much more used as two complementary models in the microbiome scene, but they don't always explain all the time series, community time series. So there is, of course, some interest in trying out alternative models and seeing if they could be useful in some contexts. But yeah, I think we have to explore this a bit more to find some examples of that. Do you have any other questions, anyone? Yeah. Don't hesitate to interrupt. Of course, this is like today, our time is very limited going through all of these things. But we have been running courses spanning over several days teaching these tools. So of course, now when you can't really try this on your own computer, unless you install all the dependencies, we have to give a demonstration, but we will be happy to provide advice and consultation on this also afterwards. So if you get interested in MiaSim or MiaTime or other tools here, do not hesitate to contact. So I will stop sharing this and then I will try to open RStudio. You can now see my RStudio, I guess. Can you confirm that you see my RStudio?
Yeah, we can see it.
So I had some technical problems with RStudio. If they persist, then I will switch to command line. But I think this is nicer interview for this. So let's see. I guess you are, many of you at least are familiar with RStudio. So this is just the interface where we can type in some commands. So I will first use this command here to load MiaSim package. By the way, is the font size sufficient so you can see this properly? I hope so.
It's okay.
Just let me know if there is this kind of problem that we will have to solve. So we will load the MiaSim library. Okay, that's done. And then Daniel also already showed how we can simulate data. So let's now simulate some time series. So we first do this random interaction matrix, and then we simulate the 4-species community. So let's look at the first row. I think Daniel went through this, but a little bit, but in order to not confuse too much, I repeat. So this part, this first line here, mat random A, if we look at what comes out, it's basically, sorry, sorry for this. This is auto-complementing so much. Okay, so as you can see, it's like matrix, 4 times 4 matrix. So this is between the species, between the 4 species, which we define here. N species is 4. So we have 4 species community. And so this is basically this describes the interactions between the different species. And we have the Lotka-Volterra model, which always relies on these interactions between the species, the pairwise interactions. So now we have this interaction matrix that we need in order to simulate instances from the generalized Lotka-Volterra model. And that happens on the next row here. So we have this simulate GLV command that is from the MiaSim package. You could even write that here to show that it is from the MiaSim package, but it's not necessary really because we already loaded the package. But we have this tool there, and then we define that we are simulating a 4-species community, And we are using this matrix for the interactions. Then we just run that line. And the TSE, that is the output, this will refer to the fact that the output from this command is a tree-summarized experiment object. And this is exactly the type of data object that I was mentioning before. So now if we print what is here in the TSE, what is included. Sorry, I enlarge this a bit. What is included? This, okay, I think it's easier this way. If I print the TSE, okay. Okay, this was my technical problem. I will switch to the command line. Just a second. Here. So we do through the same thing. So library me sim, and then I simulate the community. I basically run the same commands again. And now there was some internal error, but obviously we managed to get this object. So the TSE that comes out from this simulate GLV command, this is now the data container that we have. And if we look a little bit at the contents of this data container, you see that it has a dimensionality of 4 rows and 1000 columns. And the 4 rows represent the different species in this community. As you saw, we were simulating a 4 species community, so it's very logical that we now have 4 rows for the different 4 species. And then it has the dimensionality of 1000 because we now have 1000 time points here in the simulation by default. All right, so that's our data. And we can actually look at the contents of this by checking the counts assay. These are the counts. We have been simulating so-called counts for the data. So this is the assay. We have again 4 rows and 1000 columns here, as you can see. So I now just printed it out, and each column is 1 time series. Now, of course, we can do the normal R operation and just, for instance, look at the first column. And so that's the profile over the 4 species and the first time point. Or we can do the same for the 500 time points and so forth. So now when we have this TSE object, we can do subsetting. We can do, for instance, instead look at just the first 2 species. So we get a 2 species community instead. Or we could also limit it to 51st time points only, and then we can get smaller dimensionality for the data. So this is the data container. We can also have column data. So this is background information for the samples. In this case, we only have 1 column that has sample information. It's the time. So this is the metadata for the samples. We could also have the pH information, temperature information, and other topics like that, if it's relevant. And now when we have this object, we could start doing different analysis. And let me see if we can now visualize this so that it shows on the same screen. So I'm using the MIA-Viz package to plot the time series. So here you see the plot series command that I applied to this data, and it plots the time series. I guess you don't see the community time series now. I think I have to share you the whole screen. I guess now you can see the time series.
Yes.
Great. So this is the time series visualization that comes out of the MiaVis tool following this command. And as you can see, there is 1000 values on the x-axis. So this corresponds to the 1000 time points that we have. Now, of course, if we would like to zoom in, for instance, to the time points between 100 and 200, then we can just do this subsetting by denoting the columns here in the TSE object like this, and then we can redraw the plot. So you can see we are now having a different kind of time series, and this is because we are visualizing the only the 200 time points from 100 to 200. So if we go back to the original object without this subsetting, it looks like this. We can equally well do subsetting on the rows. Let's say, let us pick rows 1 and 2 or species 1 and 2. So it looks like this. So we can do this kind of manipulation and subsetting quite easily. If you would have to transform this, we can also transform the samples by running this this command, transform samples, and we could do CLR transformation, for instance. It happens like this. We could do logarithmic transformation like this. Now, if we look at the TSE object, we have different assays here. We have counts, CLR, logarithmic transformations. We can have different assays. And now, previously, I was showing how to pick the counts assay like this. So you see different numbers here for 4 rows and 1000 time points from the count assay. But we can also look logarithmic assay, and it looks like this. So we can now have different views on the data following these transformations. And this can be quite handy when we want to do some further analysis of the data. So you can have this all in the same object like this. So if we now would do subsetting on the objects, let's say I pick the first 2 species and I would pick time points from 100 to 200 like this. I can still have this subsetting done to all these transformed layers at the same time. And this, of course, can save a lot of effort when we do downstream analysis, and it can also help to standardize some of these so we can avoid mistakes when analyzing the data. There are many other tools available for the visualization. Maybe I could show still ordination, how to do ordination. So you have probably heard about UMAP. So I will show an example about that. Wait a moment. So this is our way to run the UMAP. So if we look at this first row on the example, what this is doing, it's running the UMAP. It's from the scatter package. So it runs UMAP. And again, it takes the same data object in itself. And then we define here that it's using the counts assay. We could also use here the logarithm assay, for instance, or the CLR assay. So we can choose what to use. And then this second line here is simply plotting the UMAP. So it is utilizing this UMAP that we calculate on the first row, and it is added on the TSE object, and then we just plot it. And now if we look at the result, it looks like this. Okay. So we can actually check if it changes somehow when we run this for the log assay instead. Just a moment. So here we run it for the log assay. Takes some time, and now it's ready, and we can print it. It looks like this. So, of course, these are just demonstration examples, but this is now all with the time series data that we simulated with the MIA sim package, and that you could do with basically any taxonomic profiling data if it was real community data or simulated community data. So, as you can see, it's relatively easy to do subsetting, manipulation, transformations, time series visualizations, ordinations with relatively little of coding once you have data in this particular object. And there are, of course, so many different examples that I could show. I will just point you to the website of the package. So let me see. So we have these different websites. So this is, for instance, the MIA-Viz website, and there is the reference. There is various different visualization tools available. For instance, this plot series command that I was just showing. So you can find that, and when you go to this site, you will have reproducible examples of this. And then also, if you go to let's say MIA-Sim website, we also have articles. We have case studies of these simulations, where you have various examples on how to use these tools and how to run different analysis and visualizations. So I think this is the brief demo that we have time to go through today. And now, if you have any questions, I wanted to leave some time for discussion, because there is quite some material. Thank you.
Thank you, Leo. And yeah, it's time for questions and comments. Anything? Okay, if there is no comments and questions.
I have a quick question.
Okay.
So I missed something, but I understand that this time series simulation you showed was not based on data, but it's just a random distribution of parameters, right?
You mean, do you mean the interaction matrix that we were using? Or... Yeah, that particular example was just a toy example, demonstration example with arbitrary 4-species community. And the main idea was to demonstrate that once you run these simulations, you can get the data in this particular format, and then you can utilize these downstream analysis tools. And I just wanted to show some brief examples of those.
Okay, thank you.
Here is the MIA time package website. So you can also have some visualizations here. For instance, this one that I showed on the slides, you can find this from the website for MIA time package. There are many other examples, as you can show, quite handy visualizations for microbial community time series. And maybe in the context of the Finding Phenol project that we are dealing with here, it is good to keep in mind that this particular framework also allows you to integrate multiple assays. So it was not only the different transformations that we can have, but we could also integrate functional data here. We could have metatranscriptomics, metabolomics, proteomics from the same samples, and you can integrate them into the same framework, and even utilize like multi-assay association tools, multi-omics, time series visualization, and so forth. But that's a bit more advanced, and you have to have to be familiar with the basic framework first. So I didn't want to go into that now, but in the context of finding phenol and multi-omics integration, this provides quite some capabilities. And it is not only our team that is developing, there is several other teams actually who work on very closely related tools also in the context of single cell experiment, for instance.
And is this multi-omics integration described in the web page as well?
Yes, we have actually a dedicated section for that. So we have a book orchestrating microbiome analysis. This is an online tutorial that has many chapters here. I guess you still see my screen.
Mm-hmm.
So as you can see, there are different chapters. Some of these describe how to do the time series, but there is small chapter currently for multi-assay analysis. So this shows how to do that, like how to actually construct it in the object. And then this shows certain examples about how you can analyze this with, for instance, to calculate cross correlations between time series, different, well, it doesn't even have to be time series. Of course, it can be cross-sectional data as well. And how to visualize that on heat maps. Then there is a thing called multi-omics factor analysis that I described actually 1 year ago in a workshop. And this is something that is also available in Bioconductor. This shows how you can run that for your multi-omics data and how to analyze the results from there. So these are the current examples, and we are in the process of adding more.
Great.
So if you're interested, look at this OMA book. This is the website, and